anywhere in the visual field. So they're kind of constantly reading out retinal populations and trying, and potentially giving the answer that they think that there's a spider there. So, there can be a false alarm, and because there's so many of these detector circuits that are kind of all operating in parallel for spiders, for all of the visual stimuli, the false alarm error, if it's very large for any one of those kind of detector circuits, if you add it up over all the detectors, it's going to be really horrible. So I think it's particularly important to have kind of low false alarm error rates. Okay, yeah.
Wouldn't false positives be worse though? I mean, like when you see there's lots of people that are startled by sort of small things that have nothing to do with a spider, but you would imagine that a car, for instance, that even if you jump back 100 times in a month, when there actually is a car there, it's important to jump back. So it seems like false positives, there's sort of like a balance.
Yeah, I mean, I definitely think false positives are important too. It's just that what I'm saying is that there's so many potential false alarm, events every moment that just because of those sheer numbers, it means that for any one kind of detector that's reading out a population of the retina, kind of giving a decision, it's going to have an even lower false alarm error, because if there are tens of thousands of millions of those kind of potential detections per moment, 1 in a million error rate is still going to be multiple events per second, which should be sort of overwhelming level of hallucination. So just the sheer numbers. Okay, and then the last thing I want to point out is that in general there's an ambiguity problem. So it's not just the issue that single neurons have noisy responses; they also have ambiguous responses, and I'll kind of talk about that more in the next slide. So for all these reasons, I think it's actually beneficial to have large populations that are quite redundant to kind of allow fast, unambiguous, and low error coding. Okay, and let me just mention a little bit more about ambiguity. So single ganglion cells tend to have broad tuning. What that means is you can get the same firing rate from many different stimuli. Okay, so if you flash a dot on the receptive field, it gets bigger, 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 the firing rate goes up. If you have the same dot size, you make the contrast go up, and the firing rate goes up, it saturates. You change the position of the dot, and the firing rate has some tuning curve. And so if you observe a single firing rate from the ganglion cell, you don't know whether it's a big dot at low contrast or a medium dot at high contrast, a little bit to the side of the receptive field, et cetera, et cetera. So there are a lot of these different properties that all contribute to the response, and you can't disentangle that when you look at one ganglion cell. But the idea is, with the whole population, you can tell those things apart. Okay, so the idea here is I'm going to describe this kind of first experiment where we've begun to kind of explore these issues of reading out population code. And so what we've done is a shape discrimination task. So what that means is you flash one of these 36 different shapes onto the retina for half a second on, half a second off. Record from a whole bunch of cells, in this case 162. And just a kind of important technical detail here. So when we did these experiments, our array technology wouldn't let us record from all these cells in one preparation. So this is multiple retinas, and so we don't have any noise correlation. So cells here are, in this case, have correlations that are only induced by the stimulus. But I'm happy to talk about what the noise correlations specifically are doing.
How many cells per retina? It's one cell per.
It's for single patches, it's of order kind of 20 to 30 cells. Yeah. And yeah. and then you repeat the same stimulus for all the different preparations. Okay, so these stimuli are likely to be ambiguous because basically each one of these letters is about as big as the receptive field size of a ganglion cell. Just to kind of mention, each one of these shapes have kind of 400 black pixels, and the rest of the screen is gray. Each pixel is about the size of a photoreceptor. Okay, so you expect that there's pretty high signal-to-noise ratio for discriminating these shapes, at least in the photoreceptor layer. And so now we're kind of looking at the ganglion cell layer, and so because they're all about the same size as the ganglion cell receptive field, if you just do kind of a linear averaging over the spatial profile of the receptive field, all the cells are going to give a pretty similar response. And also the shapes have the same center location, same kind of contrast and mean light level, so those kind of low level cues are not going to be useful in telling them apart. It's really the shape that needs to be resolved here. Okay, so what does some of the data look like here? Are kind of firing rate kind of profiles of 3 different ganglion cells responding to 1, 2, 3 different shapes. And so you can definitely see some selectivity here. So here, for this, let's look at this ganglion cell. So it doesn't fire at all to the X, but it fires a similar amount to these other 2 shapes. So it tells you something about whether the X is present, but this cell actually doesn't discriminate at all among upside down T and the bar. This one has a little bit of difference in firing, et cetera, et cetera. So when you look at individual cells, you see some selectivity, but of course you don't see any perfect X detectors or anything like that. There's just a little bit of selectivity per neuron. Of course, the problem that the brain really has to solve is there's, one response, 162 cells fire. This is what it looks like, time, 162 cells. These are the spike trains. And so, what the brain has to do is take this and, classify, figure out what the shape is, on a single trial. Okay, so how do you sort of solve this decoding task? Okay, so first of all, what we're going to do here is we're going to treat one of the shapes as kind of the target stimulus that we want to recognize, and we're going to lump all the rest of them together into the group of everything else, or distractor, because remember, we're not assuming ahead of time that we know that there are only 2 possible stimuli, so we're going to do this task of target versus all the rest of them as a distractor. And then, of course, we'll rotate and make each one of the shapes be the target stimulus, one at a time as we kind of go through the analysis. Okay, so what you're then going to do is you're going to kind of bin your responses and concatenate the response of all n cells into kind of a single vector for the whole population. And then the idea is the optimal decision rule for kind of, each one of these discriminations is 2 alternative. It's target versus everything else. And so the optimal decision rule is just going to be maximum likelihood. Okay, so if you observe population response vector R, you want to know what's the probability of the target stimulus given that response, compare it to probability